Let's do this again. All right. So today we're going to be covering a thing called the Agrobacterium mediated transformation. We've got a video down here that's 2 minutes and 46 seconds long, and that's going to contain that's going to be the stick. That's going to be everything that you need to kind of get started on this and have a basic level understanding. But before we go into that, let's look at what is our goal here today. So let's imagine we wanted to genetically modify a plant. And you could say, well, why do I want to do that? I like plants how they are. And I guess the answer is, what if you could get bigger, better strawberries or something like that? You know, yeah, would you? And the answer is, yeah. People have been genetically modifying plants as long as we could. Most of the time we have to do it randomly and blindly. This is what farmers do, and it's called selection, where you pick out the tomato that looks a little different from the other ones, but is more like what you want. It's a little bigger, a little juicier, and then you raise up some kids from the seeds of that, and then maybe you cross in another tomato that you like the taste of. And yeah, everything that you're doing there is genetic manipulation. It's just that you don't know which genes you're manipulating. Now, what we can do with modern science is we can actually have an understanding before we do it of what it is that we're trying to do and take a more rational approach. This can get us to a desirable result a little bit faster. And it can let us do interesting things. Like, for example, you can make potatoes that produce human insulin. And so then you can just grow a crop of this. For a long time, insulin had to come from either pigs or, it was like really expensive to make inside of a lab. But if you genetically modify the potato, you can grow a giant field of these, and they're not particularly good for eating, but you can then just squeeze them out and have vast amounts of insulin at just a tiny, fraction of the cost that it would be to produce it. And so there would be countries in the world where something like this would be very, very useful. That's the idea. People are also looking at, for example, edible vaccines. So I'm working on a project on an edible vaccine for the new coronavirus with some other scientists. And that'd be kind of neat. You could have a little, instead of going in and having a needle inject you, you could go in and eat a peanut or a little square potato. And that would be the vaccine. You'd be vaccinated now. And I don't know if you like needles or not, but I feel like people could really get behind that. That seems like a good thing. Anyway, to do any of those things, you have to first figure out how to genetically modify a plant. And that's what we're doing here. I guess the other thing that's relevant to Bowie County is that we actually have a fair amount of cotton that is grown in Bowie County. And it's all the acreage, I think, is owned by just a couple people. But these cotton farmers are exclusively growing genetically modified cotton. And it's not the cotton part that is genetically modified, it's the plant. So you wouldn't have like a genetically modified shirt that would, I don't know, be any different. But they put bacterial proteins into these plants. And the bacterial proteins are actually toxic to certain kinds of insects that are pests of the plants. And so this means you can grow that cotton without having to just spray entire fields full of pesticide. You don't lose all your cotton to weevils and various other kinds of insects that way. And so it's actually really good. It's like a natural way to do pesticide treatment, if you like. But anyway, we're not here to get into the ethics of the thing. Like, should we, should we leave things alone? I mean, I guess my point of view on that would be things are changing all the time. And bacteria are out there swapping genes around. Plants are out there swapping genes around. It's going on right under your nose. We just don't have a good way to see that it's happening until something new pops up in the world. Then we say, oh, wow, where did this come from? But it's just, something that was being rearranged in the background by nature, all by itself. And so I would say, all right, you may want to keep things the same, but you actually can't stop change. And so then the question is, I think, are you willing to accept responsibility for making that change? Or are we not capable of accepting responsibility and trying to do a good job with that? You know, like how much do you trust people? And that's a question for some other class. Yeah, you can go figure the answer out to that one however you like. But for now, we're going to focus on the mechanics of the thing. We want to know, nuts and bolts style, if you wanted to do this thing, how would you do it? Because that's what this biology class is. All the rest of that's bioethics and philosophy and whatever. That is not my area. So let's go. As you can see, I've stolen from the best. Mark Giltonen.
Agrobacterium tumefaciens is a soil-dwelling bacterium which resides in the rhizosphere and utilizes nutrients from the plant to survive.
Okay, you're going to notice that our host is going to go kind of fast and use a lot of science words. The reason why we do this is because each one of those words packs several concepts in to a very small space. It's a very efficient way of talking, but you've got to understand the lingo before that works. It's just impenetrable to an outsider, I think, otherwise. So what he was saying is that Agrobacterium tumefaciens, and let's go back here to where it shows its name. There we go. So let's break down its name. So this is a little kind of bacterium, and it lives in the soil. So the rhizosphere, anything with rhizo in it means root. And so this is the area right around plant roots. And yeah, swarming around plant roots, you will find these particular little bacteria and a bunch of other bacteria. The plant is producing and storing energy, and a little bit of that sugar or what have you is going to leak out. And so this is a good place to be. This is a place where you can routinely find food if you're a little bacterium. So its name means the agro is like agriculture. This is a farmer. So farmer bacterium, and then tumefaciens. The tuma is going to be the same root word as we have for tumor, like cancer. So, and faciens means to make. So it's the little cancer-making farmer bacterium. And you say, oh, that's weird. Yeah, don't normally associate cancer and, yeah, farms, at least in that way. So the way it works is that imagine I was going to give you a wonderful new genetically engineered strawberry plant. So if you have ever had strawberry plants in your yard, you know they're good for like 5 berries each or something, and then they die out and they're only good for 2 or 3 years, and you got to chop them down and dig them out rather and replace them. But what if you could just pick up that strawberry plant, and every time you wanted a strawberry, all you had to do was just punch it, bam, straight in the face, punch that plant, and then it would give you, oh, a brand new berry. And anytime you were hungry, you just go outside, and we'd call it farming, I guess, you know, plant punching. That's basically the idea here. This bacterium, Agrobacterium, wants to eat plants. It wants to eat the strawberry. And how's it going to make it? It's actually going to go in and force the plant to have what is basically cancer, because cancer is just uncontrolled cell division. So it's going to make the cells in the plant start dividing, even though they don't want to. They're going to have to. And then boom, all of a sudden it has made more of that plant to eat. Yeah, so it's hurting the plant a little bit in order to produce more food. So yeah, it's a weird little farmer, but it's okay. Okay, and this thing is going to work kind of like a shark, but let me show you this. I'll let him explain it, and then I'll come back in.
Sphere and utilizes nutrients from the plant to survive. However, if the plant is damaged, the agrobacterium moves to the site of injury via chemotaxis as a response to chemoattractants released by the injured cells.
Okay, so eventually every plant gets a little bit damaged. They get little cuts and bruises. There's actually a lot going on in the soil, and the soil is mostly made of little sort of jagged grains of what looks like sand, and it basically is little quartz crystals. So eventually these little cells will get damaged a little bit, and when that happens, instead of blood leaking out, you're going to get effectively plant blood leaking out. So there's going to be various things that belong inside the plant that are going to leak to the outside. One of those is going to be a molecule called acetosyringone, acetosyringone, acetosyringone. And you're going to see that printed on the screen in just a minute, and you can pause the video and copy that down.
Among the molecules released from the wounded plant tissue is the phenolic molecule acetosyringone. and this molecule binds to a receptor on the bacterium encoded by the bacterium's virulence.
Okay, so here we go. Acetoserine. So this is the actual molecular structure up here. That's the name. And here's how they're going to draw the molecules for the purpose of this particular video. So those are the little acetoserengones, and they are leaking out, and that's great. So this is just like, so imagine this is like a shark, and this is like, a surfer with a cut on his or her leg. So this is like the blood in the water, and these things just swarm around whenever they notice the blood, and they're going to go straight for that little broken plant cell, because that is their chance to actually inject a gene into the plant. So remember, plants have those big, thick cell walls, and they have them so that bacteria like Agrobacterium cannot just come along and, burrow into the plant cell and eat all the good stuff that's inside. So acetoserine from the plant binds to a protein called Vir A, virulence factor A. And there are several of these Vir genes, and they go A through G, if I remember right. But there's a couple that don't get used as part of this process, that are part of other processes. So we're going to stick to just a couple of these. I think there's 5 of them that we need to know.
It's a gene. The Vir A receptor is a paraplasmic protein which exists as a dimer.
Okay. So Vir A, as he was saying, is a receptor. So as a receptor is something that binds a ligand, or ligand as you like. And so that's just like any molecule that will stick to a protein. And the idea of a receptor is that a molecule can bind on the outside here, and then it can trigger some kind of change on the inside. Yeah, I don't know if you remember those tyrosine kinases and G protein coupled receptors that we were talking about last term, but those are both receptors, and those would both work by a similar mechanism. So each of these things is a Vir A. So let me trace one out with the mouse cursor here. There we go. There we go. So that's one. Here's the other one. And it actually takes 2 of these to come together, and they'll pinch one acetoserine molecule, like it takes 2 hands to catch a football if you are not Odell Beckham Jr. And so it said it's paraplasmic in bacteria like Agrobacterium and E. coli. Remember, there are 2 membranes. These are going to be those gram-negative bacteria. And so the paraplasmic space is the area between those 2 membranes. And so you're going to wait for the acetoserine to diffuse through the first membrane, and then boom, if it binds there, then you know this is fresh, and the bacterium can start swimming in the direction of whichever Vir A hit it. So Vir A is a dimer. Dimer means there's 2 copies of the protein that work together, that have to work together. If there's only 1 copy of the protein, you would call it a monomer. If there's 3 copies, you call it a trimer, 4 copies, tetramer, etc. As high as you can count in Greek, you can give these proteins names depending how many there are. And since there are actually 2 of the same protein together, we can be a little more specific. We can call it a homo, H-O-M-O, dimer, because homo means the same in science. Now, if it was 2 different proteins, if this was a Vir A and that was a Vir B, which is not the case, these are both Vir A's, but if it was 2 different proteins, we would call it a heterodimer, because hetero means different. Yeah, that's where these words come from. That's why these words get used in the ways that you know. So shown down at the bottom is the Vir Operon. And you remember operons, presumably from the Lenski experiment earlier in the term. Operons are little parts of the DNA that are going to get turned on as a group. And so there's going to be different functions here. Let's see, Vir A, you've already met. Vir A is the receptor. That's going to start out the process. Vir G is going to be a key, like a little master key, and it is going to go on and turn on all the rest of these genes. And then Vir B and D and E are going to take care of, causing plant cancer, however that happens. And we'll show you how that happens in just a minute.
Upon binding to acetoserine, it autophosphorylates, and the phosphoryl group is subsequently transferred to a protein called Vir G.
Right, okay. Let's break that down. So acetoserine binds, that's great. And then this protein is actually a receptor and a kinase. And do you remember kinases from last term? Yeah, hopefully. Yeah. So kinases are going to break a chemical like ATP or GTP. Those are the most common ones. And they're going to take one phosphate from the end of that. Just remember, ATP is adenosine with 3 phosphates at the end. And so they're going to break off the end phosphate, the third one, and attach it to something else. In this case, they will attach one to themselves, and they haven't shown that here, which is a little annoying, but that was the first thing he said. Well, they autophosphorylate. That means they phosphorylate themselves first, and then that fully activates them. And then once they're fully activated, they can then phosphorylate something else. So I guess they take care of themselves first, little selfish protein, and then they can give phosphates to others. That's nice. And then once VirG is phosphorylated, once it has that phosphate on there, VirG is now active, and VirG will go, VirG is the key, and the key is going to go turn on the entire rest of the car. It'll make all the other proteins.
When VirG is phosphorylated, it activates transcription of other virulence factors on the Agrobacterium's Ti plasmid.
This is the plasmid. This is where all these genes lurk. So remember, a plasmid is a circular double-stranded DNA. It's kind of like the bacterial genome, but it's like a little extra part that's separate from the bacterial genome, but would still be inside of bacteria. And plasmids are mobile in that you can get them shared. Like one bacterium will take it from another bacterium. And there are usually lots of copies of a plasmid in the bacterium, but usually only one copy of the genome at a time. And so, yeah, when bacteria die, they just release all of their plasmids, and other bacteria have special they look like straws for slurping up plasmids just from the world. This is how antibiotic resistance spreads in the world, and it's not just that. This is how bacteria share genes. And they do this all the time. They don't have to share even within the same species of bacterium, because a plasmid can kind of work anywhere. Yeah, because everything on the planet Earth uses the same genetic code. And so, yeah, all you have to do is pick one of these things up and boot it up and see what happens. So you can see over here at the side, you've got your Vir genes. This is the Vir Operon from Vir A all the way down here to Vir F. And Vir F and Vir C are going to be genes that we don't talk about. They go off and do other things. But Vir A is the receptor, and Vir G is the activator. G is the key. And then B, D, and E we're going to get to. So the rest of this plasmid, we've got some things that are going to act on the cellular level, and that's fine. Over here, we've got an origin of replication. This is a special sequence that's going to be on any piece of DNA, like a genome or a plasmid, that's going to be copied. And this is the binding site for those DNA polymerases. This is how they start copying at an origin of replication. And then up here at the top, this is the really important part. So this chunk between LB and RB. So LB is the left border, RB is the right border. Yeah, so yeah, great. The thing in the middle is called the tDNA, which is the transfer DNA. This is the bit that we're going to transfer. Just this bit between right border and left border will be transferred into the plant and will become genetically fused with the plant's own genome so that it becomes essentially part of the plant's DNA. The plasmid as a whole is called the Ti plasmid for the tumor-inducing plasmid. That's what it stands for. All right, let's watch a little more. Oh, we're actually, and these 3 genes, let's look at these 3 genes at the top. Auxin, cytokinin, and opine. These are all genes that are going to act as plant growth factors. So normally when a plant wants to grow, it turns on one of these genes for just a little while, and the gene does its actions. The plant grows, and then the plant stops, because they can't grow all the time. They have to store energy as well. They can't just use it. And so these 3 genes, however, are under the control of promoters that are basically always on. Again, think back to the Lenski experiment. Do you remember that promoter that was always on as opposed to the promoter that was usually off? Yeah, you probably remember this stuff. So these guys are under, always on promoters. And when we put this, plug it into the DNA of the plant cell, then all of a sudden this plant cell is going to have an irresistible urge to keep dividing just indefinitely. This is where the tumor is going to come from. This is how the bacterium causes plant cancer, which is not something that you can catch from a plant. It's just when the plant cells grow too much. Yeah, it's not harmful to you. It's only harmful to the plant and helpful to Agrobacterium.
Ti stands for tumor-inducing plasmid. This plasmid contains both the genes coding for the Vir A and Vir G proteins already discussed, as well as other virulence genes. The expression of these genes results in the transfer of the plasmid's tDNA, or transfer DNA, into the host cell. This causes the plant cells to proliferate and produce molecules that the Agrobacterium can use for energy. The first step of this transfer occurs when the proteins encoded in the virulence D genes nick and cleave the tDNA and also covalently attach to the 5' end of the tDNA strand.
Okay, let's unpack that. So now he's talking about the next protein, which is Vir D. And so they're just going to put we are going to assume that they're all Vir proteins, because they are, and we'll just have the letter on there. So remember, Vir A is the receptor up here that binds acetylserine. Vir G is the key that turns on all the other ones, B and D and E, and A and more G. Vir D comes along, and Vir D is, so it's like both scissors and tape. So it's going to cut DNA right here and right here. It's going to glue itself to the end of the DNA. And then once it gets into the plant cell, it's going to cut the plant DNA and glue its own DNA that it's attached to into the plant DNA at both ends. So that's how it's going to work. It's kind of interesting.
After the tDNA is excised from the plasmid,
So Vir D cuts both ends, cuts the right border and the left border, and takes out that tDNA, which remember has those 3 genes in there that are going to cause plant cancer, the plant growth factors. The next protein that comes along is Vir E, and Vir E is like the entourage. So imagine the tDNA is the star here, and you got your press manager out front that's making sure that they can, get access to the right places. And then you got the entourage that's going to surround and protect our stupid little tDNA.
Vir E proteins bind to the tDNA strand to prevent it from being degraded by nucleases upon transfer, and likely aid in localization to the nucleus as well. Meanwhile, the Vir B...
Okay, so yeah, the Vir E genes, like I said, are going to escort the protein. E is the escort, or E is the entourage. D basically deletes it and then glues it back in. So I don't know if that works very well as a thing to aid your memory. Then the next one is Vir B. And Vir B is a thing called a type 4 secretion system protein. And I know some of you found type 4 secretion system proteins when you were doing the project in Bio 1 with me. And type 4 secretion system protein looks like a little syringe. And so they're drawing it sort of as if it's cut in half, but it's the little syringe. And the idea is the bacteria use these to inject things into other cells. They'll inject toxins, or in this case, they'll inject actual DNA along with some proteins in order to transform this cell and cause it to grow indefinitely.
Genes are expressed, and the products of these genes form a type 4 secretion system that forms a pore between the bacterium and the plant cells.
Yeah. Okay, so there's our secretion system right there. So that's Vir B. You can think Vir B is the bridge. So Vir A catches acetylserine, A to A. That phosphorylates itself. And then Vir G, G is the key. The key turns on Vir D and E and B. And Vir D goes first, and it cuts out the tDNA. It's a little pair of DNA scissors, I guess. That's a good way to remember D. Then E is going to be our little entourage. They're going to escort our tDNA. And then B is going to act like a bridge. It looks more like a syringe, but that doesn't really, doesn't help you remember it now, does it? Yeah. So yeah, B the bridge. And then this whole thing is going to go over into the plant cell. And E is going to, in addition to escorting, E is the one that's going to direct it in toward the nucleus. So it already has the codes that are needed. It has the right protein binding sites on it that will get it sent to the nucleus of the plant cell, along with the tDNA that it is guarding.
The tDNA is able to move through this pore to the interior of the plant cell and continue on to the nucleus, where the tDNA is integrated into the host's genome. This mechanism for the transfer of DNA into a plant cell.
Yeah, so do you get now how we could use this to genetically modify a plant? Do you get which part we would replace? So you've got to keep the Agrobacterium. That's going to do all the work of swimming around and carrying your, your genes or whatever. You've got to keep all of the Vir genes, Vir A and G and B and D and E. The one thing you can change is Vir D is going to cut out anything that's between the right border and the left border. So between these two triangles right here. And so all you have to do is you can swap out the 3 genes in the middle and you can put whatever you want in there. And then the rest of this is just mechanical. It will chop out whatever you put in that DNA, transfer it to the plant, chop the plant's DNA, and actually glue in your DNA into the plant. And this is something you would do this on one plant cell at a time. And then if you have one plant cell, you can do plant cell culture, which is kind of like those embryo cultures that I think you may have done in BioLab. And you can turn one plant cell into an entire plant. It takes a little work, but it's very doable, and you just have to do it once. Because then once it's a plant, it's going to have seeds, or however it replicates, and every one of those seeds is already going to have the DNA that you want in there. And so it's going to be genetically modified thoroughly and forever. So that's the idea. And I think he's going to say that, and then we're going to be done.
Is valuable to biotechnologists for the creation of transgenic plants. If the genes on the tDNA are exchanged with genes for a desirable phenotype and genes which have products that can easily be assayed for, then desirable genes can be transferred into plants and the integration of these genes can be tested for.
There you go. That's it. So that's, a little course on how you make a 2.5-minute video turn into a 20-minute video with the magic of science. But hopefully that was helpful. Now, the original video is over here, Agrobacterium Transformation. This is your Blackboard page. And so you click that, and it's going to take you right back to that same video. And so you can watch it without my commentary, watch it with my commentary, whatever helps. So that's like our level 1 today. That's our basic level. And then our next level, kind of our ultimate goal, is to get to the point where you will be able to look at this particular paper, a review of Agrobacterium and TI plasmids, and you'll be able to understand what's going on there. So that is a much better, more detailed scientific explanation. And so if you get this question on the test, remember it's going to be an open note, open book test, open internet, in fact, and you're going to have 4 days to do it. So it's more like a little research report. It's not going to be something where you have to remember stuff, but the more that you remember right now, the faster you're going to be able to go. If you've already got a decent concept of this in your mind, I think that's really going to help you. So I would say you want to definitely watch my video, then watch the video without me, and then that's like, maybe that's level 2, maybe I'm level 1. Yeah, I think I'm always level 1. And then, have a stab at this review. Thank you very much. We'll look at the review in just a minute, but it's going to be pretty familiar, I think.